Hello, everyone at HubLE. I'm Karan Shah. I'm a postdoc here at the University of Sheffield. And I have here with me Carolyn Chlebek, and she's a postdoctoral research associate. And we're going to talk about a recent publication titled Molecular Identification of Spatially Distinct Anabolic Responses to Mechanical Loading in a Murine Cortical Bone. It was recently published in JBMR. So Carolyn, could you please tell us a bit about your study that you just published?
Sure. So essentially what we did was we applied mechanical loading to the left limb of mice and compared the gene expression in the loaded limbs and the control limbs. But what was unique about our study was we were really interested in the role of mechanical strain in the response of the bone. And so we were able to segment the mouse bone into areas of low mechanical strain, moderate mechanical strain, and high mechanical strain. And the exciting findings that we discovered were that gene expression actually varied a lot based on the mechanical strain, and the high strain sections of bone had stronger anabolic responses, more differentially expressed genes, and more activated biological processes compared to lower strain segments.
Right, that's very interesting. So just wondering, how does it compare to what happens in physiology in normal humans and stuff? Do we get areas of high strain which has more anabolic effects, and do the gene expression in those areas correspond to what you found in your study?
Yeah, so I would love to know the answer to that. Unfortunately, we can't really go in and dissect human bones all the time, right? But maybe someday we'll find some way to image gene expression without hurting anyone. But I do think that there's probably some correlates there, right? There's a lot of correlation between the mouse genome and gene expression in mice and humans. And there are certainly a lot of different bones in our body that have different levels of stress and strain across the bone based on activity and just general shape and morphology.
No, thank you. And what do these results mean for you in particular?
Yeah, so I think these results are really exciting for me because I'm really interested in mechanics and how mechanics drive biology. And here we clearly showed that higher mechanical strain does elicit a stronger biological response at the gene expression level. And it's something that I'm interested in looking into a little further.
That's great. And what was the greatest challenge you thought you found in this study?
No, go ahead.
Yeah. So I was asking that what was the greatest challenge you found in the study, and how did you manage to overcome it?
So I arrived at Cornell University, where I did my PhD as basically as a mechanical engineer. I had a lot of experience in device design and finite element analyses and almost no experience in biology. So learning how to extract RNA for gene expression was really difficult. And it took a lot of time, and my advisor was very patient with me and with all the right connections. But I think that was the biggest challenge I had to overcome to do this study.
Oh, that's great. And what was your experience in publishing in JBMR? It's one of the best journals in our field. So how do you find that?
Yeah, I thought the experience was great. We got good responses from our reviewers that certainly improved the paper. And so I felt like that was definitely a benefit in publishing in JBMR.
That's great. And where do you think you'll take this work forward? What would you want to do?
Yeah, so I really hope that this work can lead to more research in spatial transcriptomics. I think that we really showed that there are spatially distinct responses in cortical bone. And this is just in one bone, right? So who knows about the rest of the mouse? And so I think that it should set the stage there for sure. And I'm hoping too that a lot of this work also informs on levels of mechanical strain that should be investigated further for potential novel anabolic therapies that could be used in a clinical setting.
Oh, that's great. And in terms of a career, this publication must help you kind of move your career forward as well. So do you think this publication is going to help you achieve those aims that you have for your career?
Yeah, definitely. So one of my big career goals is to sort of bridge the gap between mechanical engineering and biology. And I think this paper really does that nicely and sets me up to start a career in combining those fields.
Well, that's great. Thank you for taking the time to discuss your new publication with us, and wish you all the best in the future. And we'd like to thank all the audience for looking at this video. And thanks very much, Carolyn.